Welcome to Fluent Daily, the English listening channel for advanced learners at B2 and C1 level. Every episode features a rich, flowing conversation in natural English on a topic that is genuinely worth thinking about: history, psychology, science, philosophy, and more. There are no simplified sentences here, no slow speech, no vocabulary drills. Just real English spoken naturally on ideas that matter. This is Fluent Daily. Let's get into it.
Today we have a great story, so let's get started right now.
I want you to hold your hand up and, just look at it for a second. Like, really look at the skin.
Right, just stare right at it.
Yeah. You're looking at millions of these tightly packed, super specialized skin cells. Now, take a breath. And I want you to think about the neurons that are currently firing in this massive cascade across your brain.
The ones that are literally processing the audio of my voice right now.
Exactly. The ones allowing you to understand these English words. And then think about the cells deep in the tissue of your liver. They're just silently churning away, filtering your blood, metabolizing, whatever you had for breakfast.
Hopefully something good.
Right. But here is the totally mind-bending question we are starting with for this deep dive. If every single one of those cells, like the flat, tough skin cell, the spindly electrical neuron, the chemical factory liver cell, if they all contain the exact same set of instructions, the exact same 3.2 billion base pairs of DNA.
Which they do.
Right. So how in the world do they look and act so fundamentally differently?
It really is, it's the ultimate biological paradox, isn't it?
It's wild.
If you think about it, it's like having an identical instruction manual placed in every single room of a massive house.
Okay, yeah.
And somehow, the room with the manual just knows it's supposed to become a kitchen, while the room right next to it holding that exact same manual becomes a library.
Right. Because the manual itself doesn't change. The actual sequence of letters is identical.
Completely identical.
And answering how that happens is our entire mission for this deep dive today.
Yeah.
We are exploring the hidden architecture of your biology. We're talking about epigenetics.
Yeah, the real underlying machinery.
Exactly. We're getting into the biological systems that determine exactly how, when, and where your DNA is actually read. And fair warning to everyone listening, this is going to shift your entire understanding of human biology.
It really is.
We are moving so far away from this old idea of a rigid, unchangeable genetic blueprint. And we're stepping into this reality where your DNA is a dynamic, living, breathing interface between you and the world around you.
And honestly, the stakes here really couldn't be higher. I mean, this isn't just some niche, highly specific topic reserved for, cellular biologists in a university basement somewhere.
Definitely not.
What we are unpacking today fundamentally rewrites the entire 20th century model of genetics. It completely changes how we view the physical process of aging.
Which affects literally all of us.
Right. It rewrites our understanding of how childhood adversity leaves these long-term physical scars. It even forces us to ask some deeply uncomfortable questions about how our grandparents' diets and traumas might be actively affecting our cellular health right this very second.
That part is it's heavy.
It's a complete ground-up paradigm shift.
It is. And I want to emphasize to everyone listening, we're absolutely going to get into some serious, heavy-hitting molecular science today.
We have to.
Yeah, we're going to talk about chemicals and enzymes and how DNA is packaged. But the goal is to extract those massive aha moments.
That's the light bulb moment.
The kind of insights that are going to fundamentally change how you view your own body, how you think about your daily habits, and really how you navigate the tidal wave of health advice you get bombarded with, but without feeling completely lost in all the jargon.
Which is so easy to do.
Totally. But to really appreciate how revolutionary this epigenetic machinery is, I feel like we have to start by looking backward a bit. We have to set the scene of the world before this revolution really took off.
We do. We really have to look at the classical, standard 20th century model of genetics. And, to be fair to the scientists of that era, we should acknowledge that this model was incredibly powerful.
Oh, for sure.
I mean, it gave us the structure of DNA, it gave us the Human Genome Project, it achieved amazing things, but it was also ultimately very limiting in how it viewed the human experience.
Because under this traditional model, the genome, which, just to be clear, is a fancy word for the complete sequence of all your DNA, that was viewed as the primary absolute, undisputed dictator of biological development.
Exactly. It was essentially viewed as a one-way street.
Right. I actually remember this from high school biology. The central dogma, they called it.
Yes, the central dogma.
The formula was super simple, right?
Mhm.
DNA made RNA, RNA made proteins, and proteins built the body. End of story.
The logic was completely linear. So if you had a variation in the genome, like a mutation, a different letter in the code somewhere, you produced a variation in the organism.
So maybe you get blue eyes instead of brown, or maybe you get a genetic disease.
Exactly. The sequence of the DNA letters was seen as the entirety of the information. And crucially, that information was viewed as fixed.
What, it was locked in.
Right. It was a finalized, published script that you were handed at conception, and, well, you were just stuck with it until you died.
But wait, I'm trying to put myself in the shoes of a scientist back in, say, the 1980s or 90s. Didn't they know the environment played a role back then? Like, obviously they knew that radiation caused cancer, or that certain chemicals caused mutations, right?
It did, yeah. But think about how the role of the environment was actually viewed back then. It was almost entirely seen through this negative, destructive lens.
Oh, interesting.
The environment was just something that could damage the DNA. It was an external threat. So things like UV radiation, toxins, mutagens, they could literally break the physical strands of the code or cause a typo during replication.
So it was just chaos.
Exactly. The classical view held very firmly that the environment could not systematically or purposefully alter gene expression in ways that were functional or heritable.
It couldn't rewrite the instructions in a meaningful, responsive way.
No, not at all. It was just a wrecking ball.
So the environment was just something that occasionally knocked a brick out of the wall, but it definitely wasn't the architect. Okay, let's unpack this, because this is where the massive pivot happens in the research. We move from that fixed, rigid script to this wild new field of epigenetics. And looking at the research, the literal definition of epigenetics is the study of heritable changes in gene expression that do not involve changes to the underlying DNA sequence itself.
And that distinction is the cornerstone of everything we're talking about today.
It's a huge shift.
It is a substantial, profound revision of that classical model. Honestly, arguably one of the most significant revisions in the history of modern biology.
Wow.
The epigenetic machinery is the system that regulates gene expression. It's this incredibly complex, beautiful system of chemical marks, protein complexes, and non-coding RNA molecules just kind of hovering around your DNA.
Just floating there.
Well, attached to it and interacting with it. And their entire job, 24 hours a day, is to determine which genes are switched on and which are switched off in which specific cells and at what specific times.
So I was trying to come up with a way to visualize this for myself and tell me if this tracks. Let's say your DNA sequence, that 3.2 billion base pair code, is the printed text of a massive, comprehensive instruction manual.
Okay, I'm with you.
Let's call it the How to Build and Run a Human Manual.
Catchy title.
Right. So the epigenome is all the stuff added on top of the printed pages. It's the yellow highlighters, it's the pink sticky notes, and it's those thick, redacted black, permanent marker lines that cross out whole paragraphs.
Yeah, that's good.
So they tell a specific cell, hey, read this page, highlight this. It's super important for a liver cell to know how to filter toxins. But then they say, flip over to chapter 4 and completely ignore it. Just take those pages shut, because that chapter is about making neurotransmitters, and we are a liver, not a brain.
I love that analogy. It captures the absolute most important part, which is that the printed text, the genome itself, never changes.
The letters are exactly the same.
Exactly the same in the liver and the brain. But the highlighters and the black markers, the epigenome, are constantly directing the reader. They're telling them what to pay attention to.
Right.
And here is the truly revolutionary part that just broke the 20th century model. Unlike the printed text, that system of sticky notes and highlighters is not fixed.
It moves around.
It is dynamically responsive to environmental signals throughout your entire embryonic development and across your entire lifespan.
Which means the environment, like the food we eat, the air we breathe, it isn't just a wrecking ball randomly damaging the book anymore.
No.
The environment is actually holding a pen. It's actively leaving notes in the margins based on what we are experiencing.
It is actively participating in the reading of the manual. And it goes even further than that.
Really?
In some highly documented cases in the research, which we are absolutely going to get into later, these responses, these margin notes left by the environment, are actually heritable.
They can be passed down.
Yes. from parents to offspring, sometimes echoing across multiple generations, and that happens through biological mechanisms that the classical genetics of simply passing down the ACTG sequence just cannot capture or explain.
It's mind-blowing. It really is. But I also know, and I'm sure you've seen this too, anytime a scientific concept gets this profound and this complex, it tends to get completely hijacked by pop culture.
Oh, constantly.
Yeah, like I feel like I see the word epigenetics slapped on every weird wellness product, every overpriced supplement, and every self-help guru's website these days.
It's a massive problem, and honestly, it is a vital point for us to address right here before we go any deeper.
Let's do it.
The true implications of epigenetics definitely extend across clinical medicine, evolutionary theory, and psychology. But because the core concept sounds almost magical to a layperson, this empowering idea that you can, quote unquote, change your genes with a green juice or a meditation retreat.
Right, which sounds great.
It sounds great, but it has attracted a tidal wave of popular science coverage that far outruns the actual peer-reviewed evidence base. There are a lot of highly exaggerated, frankly predatory claims out there right now.
So we need to be the bullshit filter today.
That is exactly our role. The distinction between what is robustly established by controlled scientific evidence and what is merely speculative extrapolation is critically important.
We have to draw that line.
We do. The research we're looking at is very strict about this boundary, and we have to maintain that rigor. We are sticking strictly to the molecular evidence.
I love it. Grounded in the actual science. So, to really understand how this works, to see how these sticky notes and highlighters actually function inside your body right this second, we need to zoom way in.
Down to the cellular level.
Exactly. We need to shrink ourselves down and look at the molecular machinery itself. How do our cells physically read the same code differently?
Well, let's start with the sheer scale of the numbers. Every nucleated cell in the human body has that same genome we talked about, roughly 3.2 billion base pairs of DNA.
Right. And buried within that massive sprawling sequence, there are approximately 20,000 protein coding genes.
20,000 genes. So every single cell, whether it's on my toe or in my retina, has the exact blueprints to make 20,000 different proteins.
They all have the full catalog, but obviously a liver cell, a neuron, and a skin cell are structurally and functionally entirely different universes.
Yeah, they do completely different jobs.
And they maintain those distinct identities incredibly stably through years and years and millions of rounds of cell division. I mean, a liver cell doesn't suddenly wake up one Tuesday, forget it's a liver cell, and start trying to grow hair.
Thank goodness the biology of a sudden hairy liver sounds like a straight-up horror movie.
It really would be. So the answer to how they maintain this strict identity is epigenetic regulation, the microscopic mechanisms that physically control the expression of those 20,000 genes.
Okay, so what are these mechanisms?
The research details two primary mechanisms that account for most of what we currently understand about this incredibly complex process. And the first one is called DNA methylation. DNA methylation. Okay, let's break this down so anyone can visualize it. What is physically happening on the DNA strand when we talk about methylation?
It involves the attachment of very specific tiny chemical tags called methyl groups.
Okay, what is a methyl group?
From a chemistry standpoint, it's just one carbon atom bonded to 3 hydrogen atoms.
So it's tiny.
It's incredibly tiny. But think of them as heavy physical barricades. These methyl groups attach directly to the DNA strand. Remember the 4 letters of DNA, C, T, G.
Yep.
This attachment specifically happens at the C, the cytosine base. And it doesn't just happen randomly anywhere. It specifically happens at what biologists call CPG sites.
CPG sites. What makes those spots so special? Why there?
It just refers to a specific linear position in the genetic sequence where a cytosine is immediately followed by a guanine, a C sitting right next to a G.
Oh, I see.
That specific pairing is the landing pad where the methyl group can attach.
Okay, so we have this tiny physical barricade, this little carbon-hydrogen tag, attaching to the CG pairings on the DNA. What does snapping that tag onto the DNA actually do to the gene?
It's all about location, location, location. When these methyl groups attach to CPG sites in what's called the promoter region of a gene...
Which is like the start of the gene.
Exactly. You can visualize it as the starting line, the doorway, or the on switch for that specific gene. When they attach there, it generally silences gene expression.
Turns it off.
Right. The gene itself is still physically there. The ACTG sequence hasn't changed a single letter. But because of those bulky methyl groups sitting on the doorway, the gene cannot be read by the cell's transcription machinery.
The machinery physically can't get past the barricade to read the code. It's blocked.
Yes.
Going back to our instruction manual analogy, this isn't just a sticky note. This is the heavy black permanent marker.
That's right.
The text is perfectly intact underneath the ink, but the cellular librarian trying to read the book simply cannot see the words.
That is a perfect way to picture it. And the reverse is also beautifully true.
What's the reverse?
Demethylation, which is the active removal of those methyl barricades by specific enzymes. When you remove them, it generally activates gene expression, it clears the doorway.
Well, it scrubs the marker off.
Exactly. Now, these massive patterns of methylation, millions of tags across the genome, they are incredibly important. They are initially established very early on during the very first stages of embryonic development.
When we're just embryos.
Right. When you are just a tiny clump of identical stem cells, it's the specific pattern of methylation being laid down that dictates how one stem cell differentiates into a liver cell and another differentiates into heart muscle. The methylation patterns are one of the primary architects of a cell's identity.
But if they are the architects, they don't just build the house in the embryo stage and then walk away forever.
Right.
That's where the dynamic part of epigenetics comes in.
Absolutely. While these core patterns are fiercely maintained through cell division, so your liver stays a liver, they remain dynamically responsive at the margins.
Throughout our whole lives.
Throughout your entire life. Environmental exposures, and this includes the macro environment, like your diet, your psychological stress levels, and your exposure to chemical toxins, they can send signaling cascades down into the cell that alter these methylation patterns.
They can change the barricades.
Exactly. They are constantly adapting, adding a barricade here, removing one there.
That is just incredible to think about. That eating a certain food or experiencing a stressful event sends a chemical messenger down into the nucleus to scrub off a permanent marker line.
It really is.
Okay, so that's the first major mechanism, DNA methylation, the direct barricade on the gene. What is the second one?
The second major mechanism is histone modification. And to really grasp this one, you have to visualize the sheer physical problem of how DNA is actually stored in the cell.
Okay, I'm trying to picture it.
If you took the DNA out of a single microscopic human cell and stretched it out in a straight line, it would be about 2 meters long.
Wait, what?
Roughly 6 feet of DNA.
6 feet of DNA inside a microscopic cell nucleus? That sounds physically impossible. That is a packaging nightmare of epic proportions.
It is one of the great marvels of biology. The cell has to spool it up incredibly tightly to make it fit without tangling. So the DNA strand is wound around these specialized protein complexes called histones.
Okay.
Imagine the histones as microscopic spools, and the DNA is the long thread wound around them. This entire complex, the DNA thread, plus the histone protein spools, is collectively referred to as chromatin.
Okay, I have the picture. Millions of tiny spools of thread crammed into a tennis ball. Where does the epigenetic modification part actually come in?
The modification doesn't happen to the DNA thread itself. It happens to the spools, the histones.
Oh, to the schools.
Yes. Specific chemical groups can be attached to the tails of these histone proteins. The research highlights acetyl groups, methyl groups, and phosphate groups. When these chemicals attach to the histones, it physically alters the electrical charge and the structure of the chromatin.
So what does that do?
It changes how tightly or loosely the DNA thread is wound around the spool.
Oh, I see. So it's not blocking the DNA directly with a barricade like methylation does. It's changing the shape of the packaging itself.
Yes, and the structure of that packaging is what determines whether a gene is accessible to be read.
Right, because if it's buried, you can't read it.
Exactly. If the chromatin is packaged incredibly tightly, a dense, bunched-up state called heterochromatin, the genes in that section of the thread are physically inaccessible to the reading machinery. They are silenced simply by being hidden away deep inside a tight spool.
It's like having the instruction manual, but a whole chapter is glued shut or rolled up so tightly you just can't pry the pages apart.
That's exactly it. But if the right chemical tags, like acetyl groups, are added to the histones, the spools chemically repel each other slightly. The chromatin relaxes.
It opens up.
Yes, it enters a loosely packaged state called euchromatin. Now, the DNA thread is open, relaxed, and exposed. The genes are highly accessible, and expression is permitted.
Oh.
And just like DNA methylation, these histone modifications are not static. They are fiercely dynamic, responding in real time to cellular signaling and environmental cues.
So just to make sure I'm visualizing the sheer scale of this, right? Deep inside our bodies right now, our cells are constantly winding and unwinding millions of these microscopic spools of thread. They're hiding certain genes deep in the folds and exposing others, putting up methyl barricades and tearing them down, all based on chemical signals they are receiving from our environment and our lifestyle.
You are picturing it perfectly. It is a continuous, buzzing, dynamic process of physical restructuring at the molecular level happening in trillions of cells right this second as we speak.
That completely shatters how I think about my physical body. Like, it's not a statue carved out of marble at birth. It's a living, breathing, highly sensitive machine that is constantly reconfiguring its own hardware.
That's a great way to put it.
And if these chemical marks, these methyl groups and histone tweaks, are dynamically responding to how we live, day in and day out, they must leave some kind of trail, right? Like a measurable timeline or a receipt of our biological history.
They absolutely leave a receipt. And that realization leads us directly to one of the most practically significant and honestly mind-blowing epigenetic discoveries of the past decade.
What's that?
The epigenetic clock.
The epigenetic clock. I have to admit, when I first read about this in the research, it sounded like pure science fiction.
It does sound like sci-fi.
But the data is very real. Tell us the story of Steve Horvath.
Yeah, so Steve Horvath is a highly respected researcher and biostatistician at UCLA. Around 2013, he published groundbreaking research that essentially changed how the scientific community measures the process of aging.
What did he do?
He developed the first highly accurate, widely used epigenetic clock. What Horvath did was look at DNA methylation patterns, those physical methyl barricades on the cytosine bases we just talked about, across the entire human genome.
But the genome is huge. How did he find a pattern in billions of data points?
Massive data crunching. He analyzed thousands of healthy tissue samples from people of all different ages. He fed all this methylation data into machine learning algorithms to look for patterns.
And what did the algorithm find?
Something astonishing. He found that by looking at the specific methylation status of just roughly 353 specific CPG sites across the genome, he could predict a person's chronological age with an accuracy that shocked the biological community.
When you say shocked, how accurate are we talking?
In statistical terms, the correlation between the epigenetic clock's estimate of the person's age and their actual chronological age, the number on their birth certificate, was approximately 0.96.
0.96. For anyone listening who hasn't taken a statistics class recently, seeing a correlation of 0.96 in complex human biology is almost unheard of.
It's incredibly rare. Like 1.0 is a perfect straight line. 0.96 means these 353 sites on the DNA are essentially ticking away like a flawless biological metronome.
It is an incredibly robust signal. The pattern of methylation at those specific doorway sites reliably, predictably changes as we get older. Some sites gain methyl groups, some lose them, but the overall pattern is like aging, written directly into the chemical ink of the DNA.
That is wild.
But here's the most important part of Horvath's discovery. The real value of the clock isn't that it can guess how many birthdays you've had.
Right, I already know how old I am.
Exactly. The power is that it reveals something called epigenetic age acceleration.
Okay, epigenetic age acceleration. Break that down. What happens when those two numbers, like your chronological age on your driver's license and your biological epigenetic age from the test, don't match up?
That is where the immense clinical power of the clock lies. Let's imagine a scenario. Let's say you are 50 years old chronologically.
Okay, I'm 50.
You go into a clinic, they take a blood sample, and they run Horvath's clock algorithm on your DNA. The clock looks at your methylation patterns and comes back with a biological age of 60.
Yikes.
Right. That means your biological age has outpaced your chronological age by a decade. You have severe epigenetic age acceleration.
And what does that actually mean for my health in this scenario?
The subsequent research has shown very clearly that the specific deviation predicts all-cause mortality, disease risk, and functional decline independently of how many birthdays you've actually had.
Wow. So you could take two people who both just celebrated their 50th birthday. On paper, they are identical.
Right.
But one takes the test and is biologically 40 and the other is biologically 60, purely based on the accumulation of these microscopic methylation marks.
Exactly.
And the 6-year-old biology, even though the person is only 50, faces a significantly higher risk for cancer, heart disease, everything.
Precisely. Their cellular machinery is physically showing the wear and tear, the specific dysregulated epigenetic patterns of an organism 10 years older.
I am simultaneously fascinated and terrified to take this test.
Most people are.
But here's where it gets really interesting for the listener, because the immediate, desperate next question is, What is controlling the hands of this clock?
Everyone wants to know that.
Right. What actually turns it forward faster or slows it down? If our daily lifestyle choices are sending chemical signals that change these marks, what are the actionable takeaways? Let's start with the bad news first. What are the accelerators? What ticks the clock forward?
The research has identified several very clear, very potent lifestyle and environmental factors that accelerate epigenetic aging. Smoking cigarettes is a massive one.
Or smoking.
Obesity, chronic psychological stress, a highly sedentary lifestyle, and high sugar, highly processed diets.
It's fascinating because, on one hand, none of those things are surprising. Like, we don't need a molecular biologist to tell us that smoking and sitting on the couch eating sugar is bad for us.
Right. That's pretty standard advice.
But what the epigenome does is give us the exact, undeniable molecular mechanism of how they are bad for us.
Exactly. Take smoking, for example. When you inhale cigarette smoke, you introduce thousands of reactive oxygen species and toxins into your bloodstream.
Nasty stuff.
Very. These chemicals cause oxidative stress at the cellular level. They literally damage the DNA strand.
You, like, physically break it.
Yes. And when the cell's repair machinery rushes in to fix the broken DNA, the process is messy. It often leaves behind accidental epigenetic marks like stray metal groups as a kind of molecular scar tissue.
Wow. Molecular scar tissue.
Over time, those scars accumulate, shifting your overall methylation pattern toward that older biological profile. You are literally aging your cellular hardware.
The mechanism makes it so much more real than just someone saying, smoking is bad. Okay, I need some good news. What about the decelerators? If I'm terrified my clock is ticking too fast, how do I hit the brakes?
The factors associated with epigenetic age deceleration, the things that potentially offer genuine biological protection against aging, point right back to the foundational pillars of health. Regular physical exercise is a massive decelerator.
Wait, how does moving my muscles change the methyl tags on my DNA?
When you exercise, your contracting muscles release metabolites and signaling proteins into your bloodstream. These molecules travel to other cells, enter the nucleus, and actually influence the activity of the enzymes that add and remove methyl groups.
That is wild. It's a direct chemical communication line from your bicep to your epigenome.
It really is.
What else slows the clock?
Dietary restriction, avoiding metabolic overload, and specific nutrient-dense dietary patterns, particularly the Mediterranean diet, which is strongly associated with a younger epigenetic profile.
Why the Mediterranean diet specifically?
The polyphenols and micronutrients in those foods literally provide the chemical raw materials needed for your epigenetic enzymes to function optimally.
So eating a huge plate of leafy greens and olive oil isn't just about calories. You are literally sending a chemical package of signaling molecules to your DNA to alter its methylation pattern to resemble a younger person.
That's exactly what's happening.
That is incredibly empowering.
It is the ultimate biological empowerment. It makes the epigenetic clock one of the most concrete, measurable connections between our everyday lifestyle choices and our deep biological aging mechanisms that science has ever uncovered.
It's amazing.
And I should also mention, while we are talking about decelerators, the research notes that in animal models, scientists are exploring active epigenetic reprogramming interventions.
Wait, reprogramming? Like not just eating salads, but actively resetting the clock with technology in a lab?
Yes. In highly controlled laboratory settings, scientists are experimenting with specific genetic factors, often called Yamanaka factors.
Yamanaka factors, okay.
They attempt to deliberately erase the older epigenetic marks and rewrite them to a younger, embryonic-like state. It's essentially trying to force the cells to undergo a factory reset.
That sounds like actual magic.
We'll touch on the human medical applications of that later because it's incredibly complex,
Yeah.
but it highlights just how dynamic, fluid, and potentially malleable this entire system is.
Okay, so we've established that our daily ongoing lifestyle choices, what we eat, how much we move, our chronic stress tick, this biological clock forward or backward.
Right.
But looking at the broader research, some of the most profound, deeply ingrained epigenetic marks on our DNA aren't necessarily from what we ate for breakfast this morning or whether we went to the gym yesterday.
No, they're not.
Some of the heaviest marks are stamped onto our biology during our most vulnerable early years of development. Which brings us to the profound connection between early life adversity and epigenetic programming.
This is a crucial and, frankly, a very heavy topic. We've talked previously about ACE's adverse childhood experiences.
Right.
The epidemiological research over the last 30 years is incredibly clear. Early childhood adversity, which includes things like physical or emotional abuse, severe neglect, and profound household dysfunction, strongly predicts an incredibly wide range of severe adult health outcomes.
And it's not just mental health, right? It's physical disease.
Exactly. It predicts higher rates of cardiovascular disease, autoimmune disorders, metabolic syndrome, and certain cancers.
Wow.
And it does so in a dose-dependent fashion, meaning the more categories of adversity a child experiences, the exponentially higher their risk for these negative physical health outcomes decades later in their 40s, 50s, and 60s.
It's always been one of the great tragic mysteries of psychology and medicine. Like, how does the psychosocial experience of profound neglect when you were 3 years old cross the brain-body barrier to translate into a physical heart attack when you are 50?
For a long time, we simply didn't have the biological bridge to explain that decades-long gap. How does a memory become a disease?
Right.
Epigenetics finally provides that physical bridge. The prevailing hypothesis is that early severe adversity produces lasting alterations in the epigenetic regulation of the body's stress response genes.
So the trauma rewires the system.
The trauma literally translates a psychological experience into an altered biological architecture that persists for the rest of the person's life.
To really understand how this magic trick of biology works, we have to look at the foundational research. Let's dive deep into Michael Meaney's foundational studies at McGill University.
Ah, yes, the rat studies.
This is a classic animal model study that really blew this whole field wide open. Walk us through this setup with the rats.
So Michael Meaney and his brilliant team at McGill were looking at maternal behavior in rats. Specifically, they focused on a very common, highly observable natural behavior: how often a mother rat licks and grooms her pups during their first week of life.
Okay, licking and grooming.
Right. In the rat world, licking and grooming is the primary form of maternal care and affection.
Okay, licking and grooming. That is the purely environmental behavioral variable. Some moms do a lot, some moms are more distant. What happens biologically to the pups?
They found that the frequency of this licking and grooming produced stable, physically measurable epigenetic differences in the brains of the pups.
In their brains.
Yes. Specifically, they look deep inside the pup's hippocampus, a key area of the brain heavily involved in memory formation and, crucially, stress regulation. And they zeroed in on one specific gene, the glucocorticoid receptor gene.
Glucocorticoid receptors. We should probably explain what those do so we know why this matters.
Sure.
These receptors are essentially the chemical vacuum cleaners for cortisol, the primary stress hormone, right?
That's a great analogy. Cortisol is the alarm bell. When you are stressed, cortisol floods your system. The glucocorticoid receptors in your brain detect that cortisol.
Okay.
When enough cortisol binds to these receptors, they send a signal to the brain that says, okay, we hear the alarm, the threat is over, turn off the stress response. So the more of these receptors you have, the better and faster your brain is at detecting cortisol and shutting down the panic.
So more receptors equals a highly efficient stress response. You calm down quickly, you have high stress resilience.
Exactly. So back to the rats. Meaney found that pups who received high levels of maternal care lots of licking and grooming in that crucial first week developed very specific epigenetic methylation patterns on that glucocorticoid receptor gene.
What kind of patterns?
The physical act of the mother licking the pup stimulated nerve pathways that sent signals into the pup's brain, which literally pulled the methyl barricades off the promoter region of that gene.
Wait, the licking removed the barricades.
Yes, and because the barricades were removed, the gene was highly accessible. The result: these pups naturally produced more glucocorticoid receptors in their hippocampus.
So because their mother physically nurtured them, their DNA changed its physical expression, built more of these vacuum cleaner receptors, and they grew up to be biologically highly efficient at regulating cortisol.
Exactly.
They developed a permanent biological substrate for stress resilience, all from physical touch.
It's beautiful, really. But now look at the other group. Pups who received low levels of maternal care, the neglected pups, developed completely different methylation patterns on that exact same gene. Oh no. Because they lacked that tactile stimulation, the methyl barricades remained firmly in place, silencing the gene. As a direct result, their brains produced fewer receptors.
So they couldn't clean up the cortisol.
Right. This meant a sluggish, inefficient stress regulation system. When they got stressed, the cortisol stayed in their system much longer because they didn't have enough vacuum cleaners to clear it out. They developed a permanent biological substrate for stress vulnerability.
Their early environment, just the lack of physical interaction with their mother, literally programmed their brain's physical hardware for how they would handle anxiety and stress for the rest of their natural lives.
And here's the most crucial, staggering detail of Meany's research. These epigenetic differences didn't just fade away after puberty.
They stayed.
They remained locked in, persisting deep into adulthood. And even more profoundly, they completely influenced how the female pups eventually mothered their own offspring.
Wait, walk me through that loop. So the female pup who is neglected grows up. She has fewer receptors, so she is chronically biologically stressed.
Yeah.
And because she is constantly in a state of high stress, she is less attentive and less likely to lick and groom her own babies.
Exactly. Because she is biologically wired for anxiety, her maternal behavior changes. She neglects her pups, which means her pups don't get the tactile stimulation.
Which means their methyl barricades stay on.
Which means they grow up with fewer receptors. It produced a flawless mechanism for the intergenerational transmission of trauma and stress vulnerability.
Wow.
And the most important part, it happened entirely without any difference or mutation in the underlying ACTG genetic sequence between the groups. It was purely an epigenetic inheritance driven entirely by early behavioral environment.
That is absolutely staggering. To think that a behavioral trauma can create a biological loop that self-replicates.
It's a very sobering finding.
But of course, anyone listening right now with a critical mind might be thinking, well, that's fascinating, but those are rats in a cage. Rats aren't humans. Human development is vastly more complex. Does this mechanism actually hold up in human studies?
It is vastly more difficult to study in humans, obviously, because we can't control the environment of human babies in a lab, nor can we routinely take samples of living human hippocampal tissue.
Right, that would be highly unethical.
Very. But there is a very powerful, very sobering human parallel in the literature. The research highlights the seminal work of Dr. Patrick McGowan and his colleagues, who conducted highly specific post-mortem brain studies on human suicide victims.
Just to set the context, these were suicide victims who had rigorously documented historical records of severe childhood abuse, right?
Yes. They utilized a brain bank and carefully selected brain tissue from individuals who had committed suicide and had confirmed documented histories of severe early childhood abuse.
Okay.
They compared this tissue against suicide victims who had not experienced childhood abuse and against a control group of individuals who died suddenly of other causes.
And what were they looking for?
They specifically zeroed in on the exact same biological area Meany looked at in the rats, the epigenetic methylation of the glucocorticoid receptor gene in the hippocampal tissue.
They looked for the methyl barricades on the stress gene in human brains. And what did they find?
They found significant measurable differences in the methylation of that specific gene between the abused and non-abused groups.
Oh, it was there.
It was. The individuals who had suffered severe childhood abuse showed epigenetic marks incredibly consistent with the stress-vulnerable rat model. They had more methyl barricades, silencing the receptor gene.
Oh.
It provides a direct, highly plausible biological mechanism for the long-term impact of early human adversity on the adult HPA axis, the hypothalamic pituitary adrenal axis, which is the body's master stress response system.
So the psychological trauma of childhood abuse literally left a physical molecular trace on their DNA.
Yes.
It altered how their brains structurally managed stress hormones, potentially creating a lifelong vulnerability that contributed to their tragic deaths decades later.
It's an incredibly profound finding that completely bridges psychology and biology. Now, I must temper this with the necessary rigorous scientific caution.
What's the caveat?
The expert consensus in the field requires us to explicitly acknowledge that these human studies are correlational. We are looking at post-mortem tissue at the end of a long life. We cannot definitively prove a one-to-one cause and effect in humans the way we can in a highly controlled animal model.
Right, because so many other things happen to them over their lives.
Exactly. We don't know what other environmental factors influenced those brains over 40 years. The interpretation of direct causality requires extreme care.
Right. We can't say the abuse caused the exact methylation pattern with 100% certainty because we didn't take a brain biopsy when they were 5 and then again when they were 50, tracking the changes over time.
Correct. That would be the only way to prove definitive causation, and it's impossible. However, the direction of the evidence is remarkably consistent across both the animal and human models.
It all points the same way.
It does. The safest, most robust conclusion we can draw is that early, intense environmental experience leaves durable molecular traces in the human brain that profoundly influence the biology of the stress response for decades.
It really redefines the common phrase, carrying your baggage. You aren't just carrying it in your thoughts. You are quite literally carrying it physically on the structure of your DNA.
You really are.
And exploring how that baggage is carried transitions us into what might be the absolute most mind-bending, controversial part of this entire deep dive.
Oh, this is the big one.
Because if a mother rat's behavior can alter her pup's epigenome, and that pup grows up to mother differently as a result, is it possible these epigenetic marks are actually passing between generations directly, not just through repeated behavior, but biologically inherited through the sperm and the egg?
You are entirely right to frame this as mind-bending. We are stepping into the territory of transgenerational epigenetic inheritance.
Yeah, that 5 times fast.
Right, transgenerational epigenetic inheritance. And as the literature makes very, very clear, this is the most striking, the most philosophically profound, and absolutely the most intensely controversial claim in the entire field of epigenetics today.
It feels like it breaks all the sacred rules we learned in high school biology. Like, the fundamental claim here is that acquired characteristics, experiences you acquired during your lifetime, can biologically alter your descendants.
That is the claim.
That the environment of the parent or grandparent alters the physical biology of the child through mechanisms that have absolutely nothing to do with mutating or passing on the genetic sequence itself. It sounds like Lamarckian evolution, which we were all taught was completely wrong.
It absolutely challenges the rigid boundaries of classical genetics deeply. So we have to look very carefully at the evidence because the phenomenon is genuine, even if it is currently limited in scope, and our mechanistic understanding of exactly how it happens is still heavily debated.
Let's start with the laboratory evidence, which is the most concrete because we can control all the variables. We need to look at Dr. Michael Skinner's research on chemical exposures.
Yes, the Skinner lab.
The chemical exposure study. I remember reading this. Tell us about the fungicide and the rats.
Skinner's lab was studying the effects of endocrine-disrupting chemicals, chemicals that mimic or interfere with the body's natural hormones. Specifically, they looked at an agricultural fungicide called vinclozolin.
Okay.
They exposed pregnant female rats to this chemical, but the timing of the exposure was the critical variable. They didn't just expose them at random. They exposed them during the very specific narrow window of embryonic development when the gonadal sex determination was happening in the male pups growing inside the mother.
Okay, let me make sure I have this timeline right.
Yeah.
The male pup is a tiny embryo inside the mother.
Right.
His reproductive system, his testicles, are just starting to form and differentiate. And right at that exact moment, the mother is exposed to the chemical fungicide.
Exactly. That is the critical window where the germ cells, the cells that will eventually become his sperm, are developing. What they found was that this targeted exposure produced severe epigenetic changes, abnormal methylation patterns, in the sperm of those male pups when they grew up.
Okay, so the pup was directly affected.
Yes, but here is the transgenerational bombshell. Those epigenetic changes in the sperm led to reproductive abnormalities and diseases, not just in their immediate offspring, but in multiple subsequent generations.
Wait, let me stop you. Multiple generations.
Yes.
So the chemical exposure is long gone. The great-grandson rat, 3 generations down the line, was never exposed to the fungicide. His father was never exposed. But the great-grandson's biology is altered and diseased because of what his great-grandmother was exposed to.
Yes. The reproductive abnormalities, including prostate disease and kidney issues, persisted in the lineage well beyond the direct exposure period.
That is insane.
And this finding is not just a bizarre one-off anomaly. It has been rigorously replicated by other research groups around the world and extended to other environmental compounds like plastics, pesticides, and even jet fuel.
Jet fuel.
Yeah. It establishes a fundamental biological reality. Under certain conditions, some epigenetic marks in the germline, the sperm and the eggs, can survive the reproductive process and be passed down to future generations.
That is heavy. The idea that a chemical I am exposed to today could give my great-grandson kidney disease is terrifying.
It is a lot to think about.
But again, to play devil's advocate, that's in a highly controlled lab with a massive, specific chemical dose injected into a rat. Do we have any evidence that this kind of generational echo happens in human beings outside of a laboratory?
We do, though we have to rely on epidemiological evidence rather than experimental evidence for obvious ethical reasons. We have to look at the famous Swedish Överkalix cohort studies, primarily conducted by Dr. Lars Olof Bygren and his colleagues.
The Överkalix studies. This is the fascinating story about famine and plenty. Set the scene for us. What is Överkalix?
Överkalix is a very small, highly isolated rural parish in northern Sweden. In the 19th century, because it was so geographically isolated, the population was incredibly susceptible to the extreme weather of the region.
Because they couldn't just import food.
Exactly. If the harvest failed, you starved. If the harvest was good, you feasted. But crucially, because it was a well-organized parish, they kept meticulous, flawless historical records.
They recorded agricultural harvests, food prices, birth records, death records, and causes of death going back generations. Bygren realized this was a perfect natural experiment.
To see the effects of the food supply over time.
Right. He used these records to study the long-term generational effects of extreme fluctuations in food availability. But they looked at a very specific developmental window, the slow growth period just before puberty.
Why focus on that specific window? Why not look at what they ate as babies?
Because the slow growth period, roughly ages 9 to 12 in boys, is the precise biological window when the cells that will eventually become mature sperm are developing and maturing in the body.
Oh, I see.
They are highly active and dividing at that time. Bygren wanted to know, did the massive fluctuations in food supply experienced by a grandfather during his specific pre-puberty window mathematically affect the health of his grandchildren decades later?
And what did the historical parish records show when they ran the numbers? They found striking, highly statistically significant associations. If a grandfather had abundant food, if he experienced a massive gluttonous harvest season during his pre-puberty slow growth period, his grandsons had a significantly higher mortality risk.
Wait, what?
Interestingly, not his granddaughters, but his grandsons. They died younger, often from cardiovascular issues or severe diabetes.
Wait, if the grandfather ate well, the grandson died younger? That seems entirely backward to how we think about health.
It is counterintuitive, but the data is there. Conversely, if the grandfather experienced severe famine and starvation during that specific window, the grandsons seem to inherit a biological protective effect, showing a significantly lower mortality risk from cardiovascular disease.
So a grandfather's acute metabolic experience, whether he was freezing and starving or feasting on a massive harvest before he even hit puberty, affected the cardiovascular lifespan of a grandson he might never have even met.
Exactly.
And this can't be explained by just regular genetics, like maybe they just had bad heart genes.
It cannot be fully explained by standard genetic transmission because the ACTG sequence doesn't mutate based on how many potatoes you eat in winter.
Fair point.
Nor can it be explained by a shared, ongoing family environment, given the decades and generations that pass between the exposure and the outcome. It strongly, powerfully suggests a transgenerational epigenetic mechanism.
So the famine or the feast changed the marks.
Yes. The extreme metabolic experience altered the epigenetic marks on the developing germ cells in the grandfather, and those altered marks were passed down, influencing how the grandson's body managed metabolism and cardiovascular health.
What's fascinating here is, how does that actually work mechanically? Because I thought I read somewhere, and correct me if I'm wrong, that when a sperm meets an egg and an embryo is formed, the whole epigenetic slate gets wiped clean so the baby can start fresh.
You've hit on the exact massive biological hurdle that makes this entire field so controversial among geneticists. What you are referring to is a process called global epigenetic reprogramming.
Reprogramming.
Normally, during fertilization and the very early stages of embryonic development, there is a massive, highly active erasure of the methylation patterns on the DNA. The epigenome is essentially wiped clean, scrubbed bare, and reset.
Why does it do that?
This is biologically necessary. So the embryo has totally unspecialized stem cells that can develop into all the necessary cell types of a new human.
It's like doing a complete factory reset on your phone before you give it to a new person.
Yeah.
You wipe all the old apps and settings.
Exactly. The classical accepted view was that this factory reset wiped out all environmental marks between generations. Transgenerational inheritance, therefore, implies that some of these marks, like the ones from the fungicide and the rats or the famine and the Swedish grandfathers, somehow evade this scrubbing process.
They survive the global wiping.
They do.
They sneak through the factory reset. They hide in a hidden folder on the hard drive.
Yes, that's exactly it. Biologists call these imprinted or escapee genes. But the burning questions remain. How exactly do they survive? What physical mechanism protects them from the erasure enzymes? Which specific marks survive and why?
And does this happen all the time?
Right. How reliable is this transmission in a chaotic natural environment outside of a lab? The honest, rigorous scientific answer right now is that we don't fully know yet. The mechanistic details in humans are substantially less clear than in the controlled animal models.
So where does the broader scientific community stand on this right now? If I talk to a geneticist today, what are they going to tell me?
The current consensus is that transgenerational epigenetic inheritance is definitively, observably real in some specific contexts in animal models. We can see it, measure it, and replicate it.
Okay, so it's real.
It is, and it is highly biologically plausible in humans, supported by strong epidemiological data like the Uver-Kalick study. However, the concrete evidence for its scope and its everyday significance in human health is still considered preliminary.
So we need more data.
We do. It requires substantially more molecular research before we can draw strong, definitive clinical conclusions about exactly how much of our daily health is dictated by our ancestors' specific environments.
But even with the caveats, just imagine the sheer philosophical weight of this for a second.
Yeah.
Even if it's just partially true. If true, it means our cellular machinery carries the physical, chemical echoes of our grandparents' environments.
It's incredible.
We aren't just inheriting their eye color or their height. We might be inheriting the biological adaptations their bodies made to their specific historical struggles, their famines, their traumas, and their triumphs.
It is a profoundly moving concept. It connects us to our lineage in a physical way that we never imagined. And this merging of environmental experience and biological inheritance forces us to rethink the oldest, most fiercely debated philosophical question in human biology.
You mean the nature versus nurture debate? We've been arguing about this since the ancient Greeks.
Exactly. For centuries, philosophers, psychologists, and biologists have argued about nature versus nurture. Are we a predetermined product of our inherited genes, or are we a blank slate, a product of our environment and upbringing?
Right.
Epigenetics comes along and reveals that this entire dichotomy, this whole way of framing the question, is a fundamentally false premise.
A false premise. Because the debate implies they are two completely separate, opposing forces fighting for control over who we become, like a pie chart, where we have to figure out if it's 60% genes and 40% environment.
Right. The debate always implicitly assumed that genetic determination, nature, and environmental influence nurture were independent, separable causes. Epigenetics shows us at the molecular level that they are not independent at all.
They're constantly talking to each other.
Yes. Genes and environment interact dynamically, continuously, every second of your life. And the epigenetic machinery provides the literal, physical, chemical interface where this interaction happens.
So the simple form of the debate asked, is this specific trait, let's say intelligence, or a predisposition to anxiety caused by genes or by the environment?
And the epigenetic answer is that this is simply the wrong question entirely. It's biological nonsense.
Wow.
Almost all complex traits are produced by the intricate interaction of both. The genetic sequence variation establishes the range of possible developmental outcomes. It sets the absolute boundaries. If you don't have the gene for making wings, the environment can never make you grow wings.
Obviously.
But the epigenetic regulation, actively responding to the environment, determines which of those specific outcomes within the range is actually actualized.
Okay, let me try to translate this into a core takeaway. The genome does not specify outcomes. It only specifies a range of potential responses to environments.
That is exactly the perfect way to phrase it. Think of the genome as a massive, complex menu of possible responses. The environment is the customer reading the menu, placing the specific order via the epigenetic highlighters and markers.
So to completely reframe the whole debate for anyone listening, the nature-nurture debate historically asked, is it genes or environment? But the reality is, the epigenome is the molecular record of the environment's influence on the genome. It is the physical, chemical interface where nurture actually becomes nature.
Beautifully synthesized. It is where experience becomes biology. Now, the rigorous research does clarify one important thing. This new understanding doesn't mean that everything is equally heritable and equally environmentally modifiable.
Right.
It doesn't mean the distinction between genes and environment is entirely meaningless. Some traits, like Huntington's disease or cystic fibrosis, are highly, strictly genetically determined regardless of environment.
The mutation is just too strong.
Yes, but for the vast majority of complex human traits, behavior, metabolic health, mental illness, longevity, it means the mechanisms are intimately, inextricably intertwined. We can no longer treat the genome and the environment as independently acting causes. We have to look at the messy, dynamic interface between sequence and expression.
This is a profound philosophical reframing of what it means to be human. But it isn't just theoretical philosophy to ponder over coffee, right?
Right.
Because this new molecular understanding is actively shaping the immediate future of clinical medicine. How are doctors and researchers actually using these sticky notes and highlighters to treat disease right now?
We need to evaluate the clinical reality carefully here. Where is the actual hope? And where is the premature hype? The medical applications of epigenetics are substantial and growing rapidly, but as the literature warns repeatedly, they are at an earlier stage of clinical development than the popular media would have you believe.
So we aren't quite at the magic epigenetic pill phase.
No, definitely not.
Let's talk about where it is working right now today. Where are the most advanced proven clinical applications?
The most advanced clinical applications, by far, are in oncology, the treatment of cancer. When researchers look at cancer cells, they typically exhibit profound, catastrophic epigenetic dysregulation.
So their epigenome is just broken.
It's not just that the cancer cell has a genetic mutation. It's that the entire epigenetic control system has completely crashed.
Failures like what?
Yeah.
What exactly happens to the highlighters and markers when a normal cell turns into a malicious cancer cell?
We consistently see two major simultaneous patterns of failure. First, we see widespread hypomethylation. Hypo, meaning too little. The methyl barricades are mistakenly removed from regions where they absolutely shouldn't be.
And what happens when they get removed?
This accidental removal ends up activating what are called oncogenes genes that promote aggressive cell division and tumor growth.
So the grow and divide rapidly genes, which are normally locked down tight in an adult cell, suddenly lose their off switches. The barricades fall down, and the cell starts multiplying out of control.
Exactly. But it gets worse. Simultaneously, we see targeted hypermethylation, hyper meaning too much. Excessive heavy methyl barricades are placed on tumor suppressor genes.
The protective genes.
Yes, the protective genes whose entire job is to detect out-of-control growth and force the cell to stop dividing or self-destruct. Those protective genes are entirely silenced by the heavy markers.
That is a deadly perfect storm. The gas pedal for growth gets stuck all the way down because of hypomethylation, and the brakes are completely removed because of hypermethylation.
Precisely. The cell is trapped in a state of runaway replication. But because researchers understand this specific mechanism, modern medicine has developed targeted epigenetic drugs to attack this dysregulation. Specifically, they use DNMT inhibitors.
Okay, let's pause. DNMT inhibitors. Break down that acronym. What is the drug physically doing when it hits a cancer cell in a patient's body?
DNMT stands for DNA methyltransferase. That is the specific enzyme, the biological stapler, if you will, whose job is to attach the methyl barricades to the DNA.
The stapler. Got it.
The cancer cell is maliciously using this DNMT stapler to silence the tumor suppressor genes. So doctors administer a DNMT inhibitor, a drug that physically binds to and blocks the stapler, so the drug handcuffs the stapler. Then what happens? When the cancer cell divides to make a new daughter cell, it tries to copy its epigenetic marks, but the stapler is broken by the drug. So the new daughter cell inherits the DNA, but it doesn't get the heavy barricades on the tumor suppressor genes.
Oh, wow.
The protective genes wake up, they read the environment, they realize they are in a tumor, and they trigger apoptosis programmed cell death. The cancer cell essentially realizes it's cancer and it kills itself.
That is incredibly elegant. Are these drugs actually being used in hospitals right now?
Yes. Drugs like azacitidine are FDA approved and widely used, primarily for specific hematological cancers, blood cancers like myelodysplastic syndromes and certain leukemias. And they are currently in intense development and various stages of clinical trials for solid tumors like breast and lung cancer.
That offers incredible hope for oncology. It's a totally different way of fighting cancer than traditional chemotherapy, which just tries to poison the fast-growing cells. What about other areas, the future frontiers of medicine?
There is immense financial investment and genuine scientific excitement surrounding potential epigenetic interventions for a whole host of issues: Alzheimer's, metabolic diseases, and psychiatric conditions like severe depression.
What about the aging stuff we mentioned earlier?
Right, the epigenetic reprogramming approaches. If those cellular reprogramming techniques can be developed safely, and safety is a massive hurdle because if you reprogram a cell too much, it forgets its identity and becomes a teratoma, a type of tumor.
Oh, that's not good.
No, it's very bad. But if they figure it out, it would be one of the most conceptually exciting medical interventions in the history of humanity.
The sci-fi idea of taking a pill or getting an IV that selectively resets your epigenetic clock and physically rejuvenates your organs.
Exactly. But we must reiterate a crucial, sobering point from the scientific consensus. The timeline from a compelling, miraculous laboratory finding in a petri dish or a genetically engineered mouse to a safe, effective, FDA-approved clinical application in human beings is consistently very, very long.
It takes decades.
The epigenetics field is not exempt from this agonizing pattern. We are years, likely decades, away from off-the-shelf, targeted epigenetic reprogramming drugs for the general public.
So, bringing this all down to earth, what does this all mean for the listener right now today? If the miracle anti-aging drugs aren't ready, what do we actually do with this profound information?
I think the practical implications for individuals right now are actually incredibly grounding and perhaps a bit ironic.
Hope so.
The sensationalized popular coverage implies we need exotic new interventions, biohacking protocols, and expensive therapies to control our epigenome. But the reality of current validated epigenetic research aligns almost perfectly with the most traditional, boring lifestyle advice you've ever heard.
There is a beautiful, hilarious irony here. The practical daily application of this cutting-edge 21st century molecular science, this stuff that is rewriting textbooks, is basically go for a jog, get 8 hours of sleep, stop stressing over your email, eat a diet with high plant diversity, and avoid cigarette smoke.
It's exactly that. The revelation of epigenetics doesn't substantially change what you should do for your health. The recommendations haven't changed since your grandmother gave them to you.
Right.
What epigenetics provides is a new, incredibly compelling, scientifically undeniable mechanistic reason for why you should do it. When you eat broccoli or go for a run, you aren't just vaguely being healthy or burning calories.
You are actively, chemically curating the epigenetic marks on your DNA.
Exactly. You are turning off inflammatory genes and turning on protective ones.
You are actively managing your cellular instruction manual in real time. And to navigate this new landscape of medicine and wellness effectively, the research points out that we really need to understand the language being used to sell it to us, because it's everywhere now.
This is a vital point for modern scientific literacy. Epigenetic vocabulary is bleeding out of the university labs and straight into health journalism, corporate wellness content, and popular culture. You hear terms like methylation, chromatin, gene expression, and epigenome on podcasts, in magazines, and in advertisements.
And unfortunately, half the time, they're being used by an influencer trying to sell you a $200 epigenetic resetting super powder.
Exactly. And that's why empowering yourself with the basic structural vocabulary is essential. It helps you spot accurate science versus wellness marketing fluff.
It's self-defense.
It is. Understanding the conceptual distinction between your genome, the fixed sequence you can't change, and your epigenome, the dynamic expression you can influence, is the shield that protects you from inaccurate, predatory claims.
So knowing these foundational terms, DNA, transcription, methylation, the idea of the barricades, acts as a defense against information overload and pseudoscience. If someone on the internet says, drink this tea, it will rewrite your genetic sequence, you immediately know they are either deeply misinformed or lying.
Right, because T doesn't change your ACTG sequence.
But if they say this lifestyle protocol may positively influence your gene expression by reducing inflammatory methylation marks, well, that's at least scientifically plausible and worth investigating.
Precisely. For anyone navigating the modern health and wellness landscape, familiarity with this vocabulary isn't just trivia for a dinner party. It is a fundamental personal literacy requirement to protect your health and your wallet.
So let's synthesize this incredible journey we've been on today. We started with the old, rigid model, viewing our DNA as a fixed, unchangeable destiny, a blueprint locked away in a vault, entirely dictating who we are.
And over the last hour, we have completely dismantled that idea.
We really have.
We have replaced it with a model of DNA as a highly dynamic, deeply responsive, interactive system. It is a system that actively listens to the world around you. It listens to the chemical breakdown of your diet. It listens to the cortisol spikes of your stress. It listens to the mechanical tension of your physical activity. And it listens to the broader social and emotional environment you exist within.
And it doesn't just listen. It leaves permanent notes. The epigenome is the highlighter and the black marker, furiously writing the ongoing story of your life onto your genes, and sometimes, in profound ways, passing that story down to the next generation.
That's right.
We've seen how this microscopic chemical mechanism drives the aging clock ticking inside every one of your cells. We've seen how it explains the long physical shadows cast by early childhood trauma, bridging the mind and the body. And we've seen how it completely dissolves the ancient simplistic debate of nature versus nurture.
It is the ultimate physical interface between what you experience in the world and who you become at a cellular level.
Which brings me to a final thought I want to leave you with today, a thought to mull over later tonight, long after this deep dive ends. We've established that your genome, your DNA sequence, only specifies a menu of possible responses to the environment. And the epigenome is the physical memory of those responses being chosen.
Yes.
So I want you to ask yourself, what response are you programming into your cells right now, simply by the environment you are choosing to create for yourself today? Whether it's the quality of the food you eat, the chronic stress you tolerate, the relationships you nurture, or the sleep you prioritize. Your biology is constantly listening, and it is taking notes. What are you telling it?
A powerful, necessary question for all of us.
Thank you so much for joining us on this deep dive into the architecture of life. Keep questioning the world around you, keep exploring, and we will see you next time.